Hello, I'm Chris Carberry, CEO of ExploreMars. Welcome to the first Humans to Mars webinar, Life on Mars, What Are the Implications? I hope everybody is healthy and safe in these strange days. Like many other organizations, we're launching numerous online programs to educate, entertain, and inform people interested in Mars explorations. Thanks to all of you who were able to attend our first weekly virtual Drinks with Explorer Mars. A fun time was had by all, and I hope you'll join us again next Tuesday. We're going to be doing them every Tuesday evening at 8 o'clock Eastern Daylight Time. Also, in partnership with our president, Janet Ivory, we're also hosting Janet Planet's online astronaut academy, weekdays at 10 o'clock a.m. Eastern Daylight Time. Students from around the world are taking part in this spectacular event, so you should check it out. And for today's panel, which is the first of the ongoing series of weekly webinars that will be running and probably beyond the Humans to Mars Summit, but certainly up until the Humans to Mars Summit that's taking place on August 31st to September 1st at the National Academy of Sciences building. Of course, you can find information on that at ExploreMars.Org. Our next webinar will take place on Thursday, April 9th at 4 p.m. Eastern Daylight Time, and will focus on managing isolation in space and on Earth during our current crisis. The panel will be moderated by television news personality Sonia Gavankar, and we'll have special guest Scott Kelly, former NASA astronaut and author of Endurance, Dr. Susan Ip Jewell, president and co-founder of the Mars Academy, and John Hanasek, CEO and co-founder of Avatar Medic. Should be a great and timely panel. I'd also like to thank a few of our sponsors and partners of the upcoming Humans to Mars Summit, including Aerojet Rocketdyne, Lockheed Martin, Northrop Grumman, ULA, the National Institute of Aerospace, and the Science and Entertainment Exchange, and many others, all of whom will be highlighted frequently throughout this webinar series. Now, without any further rambling by me, I would like to introduce our moderator extraordinaire, the host of Planetary Radio, Mat Kaplan. Matt, welcome and thanks for leading our fascinating discussion today.
Oh, it is such an honor and a pleasure. Thank you very much, Chris. And Chris, I don't know if you mentioned, you are the founder and CEO of Explore Mars, which is the parrot, the guiding hand for this event today. Explore Mars with the Humans to Mars Summit every year. That I so look forward to, and I sure look forward to the return of that wonderful gathering in Washington, D.C. But since we are all faced with these hopefully quite temporary necessities of making life go on from home, we are absolutely delighted. We are thrilled that all of you could join us for this first outing. I am with the Planetary Society, where I have hosted and produced Planetary Radio for the last 20 years. But we have had a wonderful time collaborating with our sister organization, Explore Mars, for a good part of that period, and are delighted to continue that today. And I bring you greetings and regards from our CEO, Bill Nye the Science Guy. Having said that, we should jump in because we have a lot to cover. I mean, it's as big as the red planet, our discussion today, and what we may find there. Let me set the stage for today's conversation with a hypothetical. We've jumped forward to 2024. A rover named Perseverance, that we'll be talking more about momentarily, has been rolling across Mars for 3 years. Another visitor from Earth, a more recent arrival, named Rosalind Franklin, has been pulling up samples from below the surface of Mars. NASA and the European Space Agency have just announced a joint press conference that is going to reveal images and other data indicating that we just may have found strong evidence of life. Will this news, as my boss Bill Nye likes to say, change everything? What do we do next, and how will it affect plans to send men and women to Mars? But I'm getting ahead of myself. So hold that thought for now as we meet the two very distinguished guests that will be joining me here. You can see them already, I hope, on your screens. I really can't think of anyone else I would rather have on board to talk about this topic, beginning with Dr. Penelope Boston. She is the Senior Advisor for Science Integration at NASA Ames Research Center in Silicon Valley. And Penny's personal expertise is the geomicrobiology of extreme Earth environments, especially caves and mines, and astrobiology. As a lifelong human space exploration advocate, she co-founded the Case for Mars conferences in the 1980s and 1990s. That makes her a founding member of the Mars Underground as well. She's a member of Explore Mars. She looks pretty great in a fake spacesuit. She earned her PhD in microbiology at the University of Colorado Boulder. She was till recently the director of the NASA Astrobiology Institute based at Ames, and is a former professor and chair of the Department of Earth and Environmental Science at the New Mexico Institute for Mining and Technology. Also, the former associate director of the National Cave and Karst Research Institute. Welcome, Penny.
Thank you. Great to be here.
Dr. Jim Green is the NASA chief scientist. He received his PhD in space physics from the University of Iowa in 1979 and has worked for NASA ever since. That included 12 years as the director of the Planetary Science Division at NASA headquarters. Under his leadership, more than a dozen planetary missions were successfully executed, including the Curiosity rover and InSight lander, both of which are still quite active and delivering great science on Mars. Jim is NASA's representative on the COSPAR Planetary Protection Panel, where planetary protection guidelines are created and agreed to internationally. COSPAR, that's the Committee on Space Research. In 1915, you're not that old, Jim. And in 2015, Jim coordinated NASA's involvement in one of my favorite movies, The Martian. Talk about infecting a planet. By the way, he also hosts the excellent NASA podcast called Gravity Assist. Welcome, Jim.
Thank you very much, Matt. I'm delighted to be here.
So let's back up a little bit and talk about the current status of exploration on Mars now and what's going to be happening there, well, in less than a year now with the arrival of this new rover. Tell us, what's the status of Perseverance?
Well, Matt, Perseverance is actually moving forward. It's all down at the Cape. And in fact, the radioisotope power system is already connected. It's checked out in many ways. It still has a number of things to do yet, but we're making great progress. So it is on track for a July 17th open window for us to be able to launch. And that's when that highway opens up for the next 3 weeks that can get us to Mars.
This is like almost everything NASA sends to around the solar system nowadays, an international effort, right?
It is, and indeed. And in fact, if you had my little graphic that shows Perseverance, this particular rover has got a fabulous set of instruments where we have participation from Spain on a weather system. We have Norway having a ground penetrating radar. We have the French working with us on some really great camera and laser systems. And we have an Italian retroreflector. But the whole point of this particular mission is really at the end of the arm. This is where we have high resolution images that we want to be able to really look at the rock record and then make decisions in terms of creating cores. It's got a core at the very end of it. And I have an example of one that I can show everyone what these cores look like. They're like a piece of chalk in size, or for those that are young enough that don't really understand what chalks and chalkboards were all about in their education, like a Crayola crayon, one of the large sizes. Once those cores are made, they're stuck indeed into an aluminum sleeve and then dropped on the surface for then later pick up and bring back. So by the end of that decade, the 2020s, we hope to be able to have these samples back in our BioLevel 4 facility and begin the process of really looking through them to determine their viability and allowing scientists to get access to them for further research. So we're doing great.
So, and even though she resembles her sister on Mars, Curiosity, You've given at least a couple of examples in that image and with those cores especially of the new capabilities that Perseverance is bringing to the red planet. Pretty powerful machine, isn't it?
Oh, it absolutely is. And the rock record is so important to us. You know, we know that Mars was a blue planet early on in its history. It went through rapid climate change. It was a blue planet during the time we know Earth had life started on it. And so maybe life started on Mars too. And so we're really quite excited about going there and interrogating that, being able to bring back those samples and look at them. You know, for all the minerals we have here on Earth, there are hundreds of minerals for which life is such a key part of that create those minerals. And so if we can find the right set of information, indeed that press conference you talked about would be really lively.
So sample return, you know, I often refer to it as the holy grail of robotic exploration of Mars, and could be for other places as well. I know you've got a slide about that as well.
Yes, I do. If we can bring that up. This was how we envision right now working with our international community to be able to get those samples back, and it all requires NASA landing a Mars ascent vehicle. This is shown on the surface already in this diagram to the left. We then have fetch rovers that pick up those samples that we laid on the ground and deliver those. That's in the bottom middle. And then on the right, we launch off of Mars those samples in a container, and then that container goes into orbit, for which then an ESA satellite will come down using ion propulsion into low Mars orbit, pick those samples up, and then bring them back to Earth. So it's indeed a multinational effort, and the planning is going real well.
Penny, you're a longtime Martian. What's it going to mean to scientists like you to get those samples back here and into our nice big labs on Earth?
Well, you know, as you say, Matt, it's been a holy grail for many decades, and pretty much my whole career. I'm sure that's true for a lot of us. And the opportunity to actually get our lab facilities here on Earth to bring all of their power to bear on samples is a new step, really, in marching forward to be able to analyze Mars materials. The missions that we've had so far and the ones to come are magnificent. But, you know, there's only a certain amount that you can smash onto a spacecraft and a lander and a rover before you run out of power and mass and all of that. And so the ability to do these really in detail studies of the geochemistry, the mineralogy, the foundational bedrock materials, perhaps even some of the fine materials on Mars will give us insights into the climate history of that planet, which is very important to our understanding of climate in general on our own planet and beyond, and also potentially allow us to look at organic materials and various other things that may be significant for the potential history of any life that may have arisen on Mars.
Jim, I want to go back to you. I gave short shrift to all of the success we've already seen on Mars with 2 active spacecraft on the surface and that flotilla on orbit above the planet. Where are we in what we've learned? I mean, we found the water. Are we still following it? And what else have we found?
That's a really great question, in the sense that, you know, there's more to it than just following the water. You know, we've seen evidence of water all over the place on Mars. That's clear. We've seen what we call reoccurring slope lineae, where water may actually be pouring down the sides of craters in a seasonal way. This is during a time period, as you can see in this slide, during indeed that summer, where the sunlight shines on the crater walls, perhaps sublimates a water plug that's sitting in, holding back water in an aquifer, and then that water pours out and runs down the slopes. We've measured that, and we know it's water. Now, some of these actually may be drifts of dust and therefore producing some discoloration, but there's so much of it, and there's so many places where it's occurring. You know, we're pretty convinced that that indeed there is a significant amount of water locked inside the planet. Now, in addition to that, Mars seems to be emitting what we would call traces of life gases, you know, like methane and like oxygen.
I'm knocking on wood.
Yeah, right. And indeed, although those things can be generated abiotically, you know, for the methane, we've been observing methane from telescopes on Earth since the early 2000s. But now with Curiosity making those measurements directly over and over again, we do see that seasonal bloom for which what we call this rush of methane coming out, although it's still a trace gas, you know, parts per billion we're talking about. And that is coming from underneath the surface. Now that could be generated abiotically. It requires water, requires certain minerals and a heat source like magma, or it could be old methane from old life in the past that is also been trapped over time and is being released. Or indeed, it could be an indication of life there today in the aquifers is what we would think. So those trace gases now are extremely important. We're making fabulous measurement of those.
And oxygen you mentioned as well. And I know that there have been, both for methane and oxygen, there have been some conflicting data, hasn't there, depending on the observer?
Yeah, indeed. The oxygen observations that we just released over the last several months is a long-term trend. It's about 5 years worth of observations, and it's a surprising trend. You know, as the planet goes through its seasonal cycle, we expect during the winter, because it's so cold, that a lot of the atmospheric gases, in particular the trace gases, will collapse down into the surface. And then as it warms up, it actually then releases those. And what we're finding is an excess of oxygen during certain times. And then at other times, we see that the oxygen actually is being removed. So this has really caused quite a puzzle. We've had many scientists really working, trying to figure out what are the abiotic, in other words, the non-biological reasons that this is happening. And we really haven't come up with a good explanation. So the oxygen observations are really one we want to stay abreast of and see what Curiosity is going to find in the future.
Penny, have we seen evidence other than what Jim has just talked about that could indicate biological activity, or at least past biological activity up there?
Well, you know, so far we haven't had the opportunity to closely examine materials, getting back to our previous discussion about sample return. But what we have been able to do, of course, is hook up what we understand about extreme environments on Earth and try to tease out the parts of those environments that are relevant to Mars. It's pretty clear we have nowhere on Earth that is just like Mars. It's a very, very challenging environment. But we have challenging environments on Earth that have components that really go into that Mars picture. And so microorganisms in their vast diversity, their unbelievable diversity on this planet, have adapted to a wild variety of conditions. Many of them are the conditions that we see in the Mars surface, and particularly, of course, from my point of view as someone very interested in the subsurface, both near and deep subsurface, where there is more protection from some of the nasty things that you get on the Mars surface. So I think that the importance of the gases that Jim has just been articulating is really because the atmosphere of a planet is its breath, essentially, and the breath of life on Earth is very, very clear. We have a very complicated atmospheric spectrum, which is the sum total of all of the complex gases that life on our planet puts out. When we're looking at a planet like Mars, if we're looking at life that has been not as robust globally as it is here on Earth for a long time, that is something of a relict biosphere, then we would expect to see a much more subdued signal. But the fact that there is this oxygen variability and the fact that we see these traces of methane are very exciting, because what that might say about life is that if it exists in the subsurface, there's a certain leak rate, just like we have with spacesuits and spacecraft and Earth itself, and that we may be catching little whiffs of what's going on underneath. In any case, whether or not it's biology or it's non-biology, when you have a life-bearing solar system like ours, in my view, every planet matters to understanding that life, even if that life only occurs on one planet. And of course, we certainly hope it occurs on Mars or has occurred on Mars, as well as other places in the solar system.
Hear, hear. Penny, you have spent a good part of your life going to places most of us would not want to go, looking for how life has found a way to survive. One of those more milder locations, I remember being with you at Carlsbad Caverns, and you pointed to a spot on the wall and you said, "You see that? That's life eating copper." So it does sure look like the enormous variety of extremophiles that we find on our planet. It's got to give you some encouragement, right?
Oh, very much. And, you know, even though I'm a Mars fan for my entire life, of course, I have an eye on the other potential habitats. And I think that the fact that we have organisms that appear to be able to tolerate almost everything, including high radiation, and organisms that have been retrieved from, you know, space exposure and complete desiccation and living amongst metals that we find toxic, but they're busy using inorganic chemistry to make those into energy sources, makes me really understand the sweep of what our type of life, meaning organic carbon in a water matrix, is capable of. It's really quite extraordinary.
Life as we know it.
Yeah, life as we know it. And of course, you know, many of us consider life as we don't know it. And that life as we don't know it extends to other ways of making life, even out of organic carbon. So there are people thinking about other alternative sets of chemistry that might work on other planets. But from the point of view of Mars, my conviction is that we're really looking for carbon-based life in a water matrix, because Mars is not all that different from Earth. And certainly, as Jim pointed out, early in its history, it was much more similar. And so that you know, early childhood of our rocky terrestrial planets together seems to me very significant. And even Venus in its current state, which is quite inhospitable to life as we understand it, but its early history may have also, you know, shared in habitable zone. So, you know, it's not just Mars alone that is compelling, but really Mars as this beautiful red jewel within this entire spectrum, this entire crown of jewels that we have in our solar system, and what they offer for life.
Have your studies and what we've learned from others in looking for biosignatures, that key term, has it led you to the point where you think that if we had life staring us in the face, we would recognize it?
Well, yes, maybe. The slide that was just put up shows a variety, and this, of course, is from my own work. All of these colorful and drippy, gooey things that you see are all examples of microbes, as tiny as they are, but making major changes in the rock and mineral environments that they live in. So the orange stuff that you see that looks sort of like a pizza or the surface of Io, that's all rock breakdown material that's been chewed through by organisms that use manganese and iron rather than, you know, eating hamburgers for lunch. They eat rock, and they use those minerals, and then they poop out, so to speak, all of this fluffy stuff. And so they're major geological agents on Earth, but it shows that they can make their living off a geological source. You can see the screaming blue patch that one of my French colleagues is standing by, sort of in the middle lower part of the screen. And you can see an example of the same process. You were talking, Matt, about the copper material that I showed you. These are organisms all over the world that are different, but they're different and very unrelated, but they still are able to do the same thing. And so that makes me even more excited, because it doesn't have to be the same organisms. It's finding a pathway of organisms in different places that can use the same energy sources. And so to me, that seems like an encouraging sign for extraterrestrial life.
Hey, if you've just joined us as we hit very close to the half-hour mark, this is the Explore Mars Life on Mars webinar, the first of a series sponsored by Explore Mars. And you just heard from Penny Boston. And I'm going to go back to Jim Green, the NASA chief scientist now. Jim, I want to jump back now to that hypothetical that I posed at the outset. That press conference, which you would very likely be a part of. I hope you're still on the job in 2024. You've got big news for us.
Right.
What's this going to mean if indeed we find evidence better than we've gotten in the past? I'm thinking of a certain meteorite, ALH 84001, that seems hard to argue with, that maybe we have found at least evidence of past life on Mars. What's that going to mean to us here on Earth?
Well, let me just mention, of course, there is an important aspect of thinking about that future when we start that announcement, and that really boils down to looking at what's happened in the past, you know, by analogies. The ALH 84001 is a good one in the sense that, you know, we would organize that press conference. On that press conference, we would have the scientists that are announcing the discovery, but also scientists that are a little skeptical. And so that announcement then would go out to the public, and then there would be a period where the community of scientists would really dig into the results. Perhaps if we return samples soon after that, they would then be able to have those in the laboratory, et cetera, and really tease it out, and then public reaction. And then an education has to go on to everyone in terms of the context of what this means. But if we just take that supposition that indeed in the long run it proves to be true, and we have found that second genesis and it is on Mars, what does that mean? And here by analogy, I think we have to look at perhaps several. One comes to mind is what Copernicus did. You know, at the time Copernicus was coming out with the theory that the planets went around the sun and not everything went around the Earth, it changed the world view. Everyone mentally had to now rearrange their thoughts about their place in the solar system and the universe. And it had a profound effect. You know, people thought, well, we are the center of the universe because everything goes around us. And now they go around the sun, just like the other planets. And then to them, that means, well, maybe there are other societies like us on other planets. This was a really profound change. I think, indeed, we're going to have to come to grips with that. Many people, because of our literature and our movies, are, you know, all set to accept that there's a second genesis out there. And in fact, many scientists, and that would include me, think it's almost inconceivable that there isn't some sort of life out not only in the solar system, but certainly on other planets. And so our worldview then once again will have to change. And I think it will change many different things. The results of finding microbes might be as revolutionary as cracking DNA. You know, the concept of what's come out of microbiology, which is only a handful of decades old, has just been phenomenal. And that is awaiting us with this kind of discovery if we do indeed find microbial life on Mars, I think.
Penny, what would this mean to you and your colleagues, this announcement and the provision of this data?
Well, you know, I spent my entire life doing this, and so have many of my colleagues. So have dreamed about that happening in my lifetime. I hope it does. I'm trying to stay as healthy as possible to get the maximum chance. And, the minute we got any kind of indication that there might be extant life or even extinct life, okay, because I also do paleomicrobiology and I'm interested in the entire deep history of planets with life, the entire community would go into a frenzy of trying to test whatever features this potential life would have. And so there would be an enormous flourishing, maybe in directions that we're already pursuing, maybe in new directions. It depends on what those results would find. And then I would throw one heck of a big party.
I'd like to attend that.
Please invite me.
I will.
We should get into the topic that maybe our sponsors here at Explore Mars are most interested in. After all, they are the humans to Mars people. And that is what the possibility, evidence for not just past, but possibly existing life on Mars, stuff that's still kicking up its heels there today, would mean for sending humans there. Jim, you were a big contributor to that movie, The Martian.
Yes.
It would not be a good idea for us to head there and plant poop potatoes, would it?
No, actually, Curiosity has found the nitrogen, oxygen, carbon, phosphorus, and sulfur on Mars, all the right stuff. The soils are moist, and indeed, there's nitrates in the soils. Now, where Curiosity is sitting turns out to have alkaline soils, not acidic. So what would grow better here would be beans and asparagus. And I don't know about you, but if I had to eat 2 or 3 years worth of asparagus, I'd just take my helmet off and walk outside. But I'm assuming I could do the potatoes. I'm assuming we all are assuming there'll be more acidic soils too elsewhere. And so indeed, the analogies between Mars soils and Earth soils is really strong. And that's really quite important. If we found evidence for current life, that current life has to be below the surface. We haven't found evidence of life on the surface. And for human exploration, I think we'd have to talk about how we're going to share Mars. And I think we can share Mars. There are many approaches, unlike the Martian, where the concept was you land Ares 1, and the next time it's Ares 2, Ares 3, Ares 4. We would plan to go to one particular area, perhaps 150 or 200 kilometer area called an exploration zone, where we land in one part, live in another part, and then have the ability, mobility, to go around and perform a whole variety of scientific experiments, but really confine ourselves to that part of Mars. And in so doing, then it gives us a wonderful opportunity, perhaps over several decades of continuing to go there and continuing to build and develop things at that site, an opportunity to really learn the deep, and obtain a deep understanding of what Mars is all about. And then we can take it to the next step with that kind of knowledge. So I'm all for sharing Mars. Now, you may know that many scientists, particularly Carl Sagan, thought that if we found microbes on Mars, we need to leave Mars alone and go to another body in the solar system. But I think because life also has to evolve over time, the evolution of that life is going to be completely different than ours. And that gives us, I think, an opportunity to coexist. And those are some of the new ideas that are coming out now.
Annie, what's your view about all this? I mean, I think you want to see boots on Mars as well, but...
I'd like to see my boots on Mars, actually.
Me too.
But I'm going to be too old, I'm afraid. But, it's something I've wrestled with my entire career, because I have this desire for exploration, for our species to go beyond to other bodies in our solar system, and who knows, someday even out of our solar system in some number of hundreds of years. But at the same time, of course, I'm very aware of the deep ecology aspects of another biosphere. And this applies to Mars or any other biosphere that we may find in our solar system. And that is, how do we study it? How do we perhaps cohabit with it, as Jim points out, without doing damage to it, and in turn, without doing damage to us? I think that many years ago, probably 25, when I first started writing about the Mars subsurface as being the best place to look for life on Mars, it was not taken very seriously. We knew a lot less about the planet at that time, but we've plugged along on that theme, and I think it's become manifest that that is the place, as Jim says, where we would have the highest chance of finding subsurface life, even if Mars once was covered in life at the surface. That the subsurface, in many cases, particularly even on Earth, is a refugium, a refuge for organisms when circumstances change on the surface. We see this in microorganisms and macroorganisms. So there are caves with fish and invertebrates that have been separated for 20 million years from the surface and things of that sort. So I think that the expectation that I have is that any Mars life is going to be quite deep. And that helps us with this conundrum, because as long as we don't contaminate aquifers, as we're trying to get resources for human use, then the surface Mars environment is very harsh. And while I wouldn't call it entirely self-sterilizing, it will do a lot to reduce the plume of biological contaminants within some distance from a human colony. And then, of course, if we want to study things like the slope lineae, these trickles of water, briny water that Jim mentioned early on in our discussion, those are juicy targets, literally and figuratively. We can't wait to get our hands on them, but our hands will not be our hands. Our hands will hopefully be sterilizable robotic sampling devices that will be able to send out and bring those samples back for analysis. So there are ways to do this. And, you know, we're living through it now with this pandemic that we're all experiencing. How do we separate our activities from, in this case, a pathogen, but in the case of humans on Mars, how do we separate ourselves from potential organisms that are the Martians and keep all of us safe? And so all of these real world things are being worked out in real time. And as you know, Jim mentioned, or you mentioned that Jim is on the COSPAR panel, that dates back to the 1950s and the early part of the space age. So it's not like we just discovered that, oh, gee, there might be a problem. Many of us have served on panels and workshops, and we're writing about it all the time. So it's very, very much foremost in our minds about how to do this safely. I think we can do it. I hope I'm not being naive.
Jim, I'm really glad that Penny brought us back to that COSPAR committee that where you are the planetary protection representative from NASA, from the United States. How is this being considered by the international community? These questions of protecting Mars, but also wanting to go there and have humans explore.
Well, indeed, COSPAR over the years has looked at each and every one of the bodies in the solar system that were being considered by space agencies to go to. And indeed, early on, we didn't know much about the moon in terms of whether there would be pathogens there. And so consequently, those guidelines that came from the COSPAR committee that the international community signed up to meant that we, for a NASA perspective, wanted to implement a quarantine system. So when samples came back from Apollo 11, you know, the astronauts went into quarantine and the samples went into quarantine. And then we went through our processes of indeed interrogating those and understanding what they are, what we had, whether there were pathogens, etc., watching the astronauts over a period of time. And then finally, recognize that indeed we could have uncontained sample return from Mars. We didn't need to go through the quarantine process. And so then that means the guidelines were revised. So the committee indeed takes it very seriously that as we learn new things about each of the bodies that the space agencies are planning to go to, we take that into account to modify those guidelines. And that's been working really well. In fact, just recently, we took a good look at Phobos. And we recognized that Phobos, indeed, one of the moons of Mars, we could actually back off some of the more stringent guidelines on returning samples. And so we've categorized Phobos as a location where we can have unrestricted Earth return of those samples. And the Japanese JAXA mission is being planned to go there and has many international connections and components to it. But that required for us to bring all the knowledge we knew about Phobos forward and not consider it like Mars in a category of more restriction. And so we're going to continue to do that. And that process is working well.
We are rapidly approaching the point in this program where we're going to be turning to your questions. And I do see that lots of you are submitting them. They are being consolidated for us by the president of Explore Mars, Janet Ivey, of Janet's Planet. And by the way, I want to take the chance to acknowledge that a lot of Janet's students are watching us right now. Tapa Sweeney from India, Alex from Pennsylvania, Judah, she's not sure where he is, but Judah, welcome all of you young people, you who may actually be wearing those boots that go to Mars. We certainly hope that you'll have that opportunity and do some of this exploring for us up close and personal. Before we get to some of the questions that are coming in, I've got one more to throw at you, and Penny, you touched on it, but it's a question that Explore Mars CEO Chris Carberry posed before we started this, and that is whether what we have learned about both how we might handle isolating ourselves from life on Mars or anything else in the field of astrobiology might have helped prepare us to deal with the challenge that we're facing around this planet right now, the pandemic. I mean, do you see any relationship there?
Yes, I do. I think that one of the things that this horrible circumstance will maybe help is eventually in better public communication. You know, when you are trying to talk to people who are not microbiologists or scientists of some other sort about microbial life, it's very hard to communicate the size of these individual entities, their capabilities, and just how easily spread they are. You know, in many cases, that's very benign. There are many microorganisms that we need in our environment. They basically run a lot of the biogeochemical cycles on our own planet. So on balance, they're beneficial. We only sort of notice them on the broad scale when they're deleterious, like in the case of the COVID-19. But you can derive some lessons from this. You know, the fact that we have a new virus on the scene means even though it's related to other Earth viruses and similar to others in its group of coronaviruses, it's many different properties. And so a lot of the scrambling that's going on now in the medical and research communities is to try to figure out its properties. And so the way that we do that in real time is orders of magnitude accelerated compared to what we would have been able to do with something like this even 20 years ago. So within mere weeks, we had an entire genomic sequence for this RNA type of virus. And that was propagated around the world. And so the biggest issue is, you know, we are a world full of people with different traditions, different levels of technical sophistication, and different access to communication. And so it's the human response to that that's been the trickiest part. But one of the things that I think is important to communicate is that microorganisms, for the most part, are benign and absolutely essential to us. And so one of the things I don't want to see is an increase in sort of mindless germophobia from this. And, you know, that could be a natural outgrowth of this. But we are talking about studying microbial scale life on another planet, although we're mostly looking at bacterial size things. And if you look at a bacterium, you can probably line up maybe 80 or 100 of them across the diameter of one of your hairs. But if I were going to put viruses end to end across the diameter of your hair, I would probably put 1,000 or 2,000, right? So they're even more minuscule. So I think we're mostly focusing on bacteria size organisms. And so those are actually much easier to control and work with for the most part than the viruses, because the viruses are tinier than dust specks, whereas the bacteria are much chunkier, and so our methods of controlling bacteria and working with them in many ways are easier.
Jim, anything to add from the NASA perspective?
Well, yes. Penny did a wonderful job with explaining the various aspects of that. What's critical also to understand is, viruses aren't considered alive. They really are cellular parasites, if I were to describe them in some way, and they invade the body and they have some genetic material associated with them. But in reality, for them to live and grow, they actually have to co-evolve with a host. And so when we think about bringing material back from Mars that is alive, that life has perhaps a second genesis, perhaps is related in some way to us in terms of the fact that it started in a similar environment like Earth, but it has a completely different evolutionary track. And so the current thinking is, yes, we're going to quarantine those samples. Yes, they'll be in what we call a biolevel 4 facility, where they handle anthrax and everything else, and it will be closely monitored. But I think, as Penny points out, our ability to understand the microbial and virus environment will allow us to use those current tools to interrogate those samples and begin the process of releasing things to the science community, which will be really important to do, because that's where those laboratory equipment really comes into play. In many places around the world that will really tease out some of the most spectacular science in terms of really having a deep understanding of that planet than we can dream of today. So those are all just right around the corner, and I'm hoping also to be around when we start cracking into that next generation.
You and me and Penny, all of us, and everybody watching this program, I am sure. All right, we are now at approaching about 10 minutes of the hour. But I have also heard from both of our guests, Jim Green and Penny Boston, that they might be able to spend a couple of extra minutes of bonus time with us. Yep. So let's go ahead and start getting to your questions. And I know that many of you are submitting them through the Zoom window, but we also got a couple. Well, I'll read one of these that was submitted ahead of time to Explore Mars. And this came from Andrew Planet, how appropriate, in Gibraltar. And so we also welcome our international audience for today's program. Andrew says, if we ever find native life on Mars, and if it had the same method of storing genetic information as we all share here on Earth, would that mean that biology originated independently at both worlds, but had evolved analogously, because it was a good solution, I guess, or that they've evolved out of a common ancestral biochemical panspermia? There's a term that we all love. Penny?
It's one of the big debates that we have, and we're not sure how to make that decision yet. If and when that arises, then we'll have to do a lot of investigation. What I think most of us are hoping for is what Jim mentioned, which is a true second genesis, where the life on Mars would be so different that we wouldn't have this conundrum. But if the life resembles ours in terms of its basic biochemistry and molecular machinery, then we're going to be faced with this conundrum. Is it regional, shall we say, panspermia within the solar system? Or is it that it's such an optimum solution to make life the way that we do that it got reinvented again? And it's very hard to know how to actually go forward. And so that's where detailed work in the future would have to come to bear. I think there's no easy answer.
Jim, you're nodding.
Oh yeah, so I think the concept of, okay, we're going to look at that life, and if it is indeed RNA, DNA, has all the right stuff, we're going to want to see if there's a common ancestor. Okay, that's how that would relate to us. But as Penny points out, that's going to still be incredibly difficult, and it's really because of the fact that life has that attribute that it has to evolve, and that environment is so different than ours, on a daily basis. The temperature swing is on the surface alone is 170 degrees Fahrenheit in one day, and it's bathed in ultraviolet light, and so maybe this life has found a way to repair itself or to survive in those environments, and that's tremendously exciting. If we can tease that out and it relates to our life, then we have new medicines, new approaches. It's just a complete revolution in our ability perhaps to live and grow on other planets where there are extremes. So this is just really an exciting opportunity for us that we just can't pass up. Let's find this life and bring it back.
That's thrilling. Jim, I'm going to throw this next one to you, and it comes from one of Janet's young fans, Tapa Sweeney. I apologize, Tapa Sweeney, if that's not even close to how your name is actually pronounced. She says, how can we guarantee that we are not putting life forms, our own biology, on Mars from all the probes that we are sending to crawl around on the red planet, even though we try to sterilize the stuff that goes down to the surface?
This is a really good question, because indeed it's all about that planetary protection guidelines. We early on through COSPAR and had this international agreement that Mars would be a place where potentially life could have been or even is extant, living there today. And so consequently, those guidelines tell us that there's a level of sterilization that we want to do. We want to be able to have an opportunity not to contaminate that planet while we learn much more about what that planet is like. So we've gone through quite a program in NASA, and many of the other agencies have stated they have too, through those processes of learning how to clean a spacecraft, learning how to be able to ensure that there's minimal or nearly as close to zero bioburden, we call it. That would be types of microbial or other types of life from Earth on the spacecraft as possible. And in particular, those places that actually touch the surface. And so that whole topic has evolved over time, for which many new and exciting techniques have come up, and we're still doing original research in that area. So we plan to keep Mars as pristine as possible in terms of not contaminating it as we possibly can. And of course, once our rovers or landers are down on the surface, they too are bathed in ultraviolet light, which really takes out another layer of bioburden that occurs by us launching it and getting it there in some sterilized form. We can't completely sterilize it, but the environment is also a sterilizing environment. So I think we're overall internationally doing real well.
And I have to apologize. That question actually came from Michael. I don't know where he is. I apologize to you, Michael, and to Tapa Sweeney. Tapa Sweeney did have a question, though, and it's more a geology question about rocks on Mars. Are they similar to what we know we have on Earth? And I think the answer is yes, right? Yeah. But I mean, are there geological implications for our search for life on Mars? I don't know which of you might want to answer that.
I'm willing to jump in. I have one of the slides and the backup slides, which is my number 3, I think, for Wade out there. One of the big revolutions in thinking of the last 10 or 15 years has been the degree to which life on our planet has affected. So slide 3, which would be let's see.
Is that your last one? Biominerals?
Yes, it's
yes.
Biominerals. I'm sorry.
Oh, it's number 7. There we go.
Sorry. We're learning to do this online thing here. So the biominerals in the solar system, it's been pointed out by a guy named Bob Hazen, who's at the Carnegie Institute in Washington, and a number of his colleagues more than 10 years ago now, that if you look at the early solar system and a lot of the bodies in our solar system, like small bodies that have not had extensive geological evolution and don't appear to have life, that you got 12 primitive minerals. And on Earth, we have in excess now, actually, probably about 4,500 known minerals on Earth today. Many of them, I must say, are found only in caves. So just giving caves a plug here.
Okay.
But, in their thinking, the mineral wealth that we have here on our planet is due to abundant liquid water and the fact that we have tectonic, plate tectonics that helps to mix up the crust and down to the mantle and so forth. But the rest of that 4,400, 4,500 is due to the geochemical and biochemical interactions of life with the geology of the planet. And so one of the things that I think we're very interested in is not just the bulk chemistry of Mars, but where we find a lot of these unusual minerals on Earth are places, micro niches, sometimes hidden niches, many of them in the subsurface or at the bottom of the ocean or in rock fractures where the life is actually working to create these minerals. And so this is another whole class of biosignatures that I think we did not really grasp in its complete form until Bob Hazen and his colleagues pointed that out. And so I think this is has been a revolutionary concept. Yeah, the slide that that Wade just put up now shows the life-produced gases, which relates to Jim's early remarks today. And then the biological molecules, there are molecules that we can see from space. Chlorophyll is the most abundant protein on our planet, and it drives the solar production of biology. But there may be other pigments that we could find, and then of course there's morphology. So when you add all these things up together, biominerals, real fossils if we ever find them, microfossils, and my favorite is Baby Yoda, so I'm still holding out hope for that.
Let's see.
Well, indeed.
Go ahead, Jim.
Let me just follow up by saying, Curiosity has an experiment that actually looks at the mineralogy, and so consequently, over its 5 years of operation on the surface, it brings in material and does X-ray diffraction, and then that pattern tells us the mineralogy, the structure of how these things are put together. And right now, I think the count is well over 300 individual minerals, and of course, that we have found and discovered. And then the first thing we want to do is that a new mineral? Is that something we don't have, or do we have it? And in every case, we've said, yes, that is also exists here on Earth. But the concept then of bringing back those samples, bringing them back and actually having in the laboratory an opportunity in that rock record to look at the change in mineralogy over time, because we have cored that rock and we have that time record, is going to be incredibly important for us to understand how Mars has evolved, and evolved in a way that's different than the Earth, because of, perhaps its distance from the sun, or it's the lower mass, or it didn't have a magnetic field that kept it preserved for as long as the Earth has had. all those other factors then come in to really understand and figuring out that history of Mars. So this is really an exciting time, and that rock record is just so key to being able to tackle those questions and those things that Penny was bringing up too, all important.
Let's see if we can squeeze maybe a couple more questions in here. We
Sure.
are now into bonus time, and so we thank all of you for sticking with us for over an hour here. And this one comes from, if I can find it again, from Gene of Talking Space, another great podcast. For both of you, actually, given the fact that there is an interest in Mars colonization by several commercial entities, what would the impact of microbial life on Mars or a second Genesis due to these commercial ambitions? Jim, why don't you start with that one?
Well, you know, the planetary protection guidelines that come out of COSPAR are implemented by each of the space agencies. And so our commercial entities here in the United States would indeed fall under those same guidelines. So indeed, they are just as interested as NASA is in terms of what that means relative to the exploration of Mars. So we're working together to be able to think about how we would approach colonizing Mars in the sense of that exploration zone. Commercially, we expect them to be major partners. We expect things to be landed in those areas, things that can be put together. And in fact, they may eventually have commercial astronauts that will go. But because they are US companies, they will follow the US implementation, the NASA implementation. And that's why they're interested in these topics just as well as we are. We plan to do, you know, the right thing. We think we can, but to do that, it requires these samples coming back for us to really interrogate them, learn so much more about Mars that will allow us then to revisit those guidelines and come up with a way that makes it even easier to explore Mars.
Penny, I'm going to go right to this last one and throw it to you. It's partially personal. From Rachel Tillman. What inspired you to become part of the search for life? And really a separate question, but there's some relationship. How can we integrate everybody who's interested in this, not just NASA, into sharing these findings, making them part of the public education system, like Rachel says during Apollo, where, you know, I can actually remember schools with televisions rolled into the classroom so that we could watch the latest mission landing or return to Earth. I mean, have you thought about this, Penny? How do we share the passion that you and Jim and everybody else watching this obviously feel? Hey, Penny, you're muted there.
I think it muted me itself.
There you go.
Our computer are becoming sentient as we are all home.
Speaking of finding
Genesis, yes.
biosignatures that we might not recognize.
So, Rachel asked what inspired me. I was a very curious little kid, like most kids are, right? And I started reading science fiction very early. That was it. I wanted to get in my rocket ship and cruise the galaxy looking for things with long tentacles and 15 blue eyes, and I still do. So I think that, you know, the arts, the science, the literature, all of that comes together and inspires us. I come from a family derived from many different countries, and that has moved around a huge amount. And so there's that taste for exploration, you know, in my background in terms of Earth. But I think the joy of what we do is impossible not to communicate. I know Jim and I and many of our colleagues go out of our way to do public events, large and small. So, you know, I have done many discussions with 4th grade classes and elder hostels and gem and mineral groups and anybody that wants me to talk, if I can fit it in, all the way to, you know, big television broadcasts that are commercially produced, or PBS or whatever. And I know Jim is the same way, and he has his own podcast, which, you know, serves that function. I show up to things, and I can't keep it inside. I mean, this isn't just a job where you kind of punch in the, you know, 8 o'clock and go home at 5 and stop thinking about it, right? This is an all-consuming lifestyle, and it has saturated my entire life. And even when I eventually go off NASA's payroll, I don't plan to go off the scene entirely, you know. And that this is true of so many of our colleagues. I think, Jim, you will have seen that too. You know, they retire, maybe they're not picking up a paycheck, they're picking up a retirement check, but they're still actively doing research, they're actively thinking and writing and talking to folks.
Yes, indeed. In fact, it's going to be the case, I can see it now, when we land on the moon. Where that teacher is going to say, class, take out your iPads, and they'll all bring up, you know, the landing where the first woman, you know, and maybe even the next woman, but at least the next man will step on the surface of the moon. Now, I was alive in high school at the time of the lunar landings, and it was just awe-inspiring, even with the grainy, yucky, you know, black and white TV.
Yeah.
And so today we're going to be doing it in high definition, and the colors will be spectacular, and the shadows will be long, and it will be incredibly eerie, but it will be just as inspirational. So everyone, you know, that hasn't seen that original lunar landing of any of the Apollo astronauts have really got a treat in store for them, and that's coming up.
What a great way for us to end this wonderful conversation. I want to thank both of you. There truly were no better people to be a part of this discussion of life on Mars and why we, examples of life down here, are so excited about it. Thank you, Jim Green, NASA Chief Scientist. Thank you, Penny Boston of NASA Ames, astrobiologist.
Thank you.
And thanks to all of you for tuning in, for joining us for this, and to Explore Mars for making it all happen, and the great people there who have been helping us out: Wade, Janet, Chris, Ron Sparkman, Adrienne. It's just been terrific, and I know that this is just the first of many that Explore Mars plans to bring you. I'm Mat Kaplan, the host and producer of Planetary Radio for the Planetary Society. I hope you'll check out our podcast, Planetary Radio, and keep looking up there at Mars, because we're there, and we're going back. Thanks again for joining us. Have a great day and stay safe.
Live long and prosper.
Live long and prosper. Thanks, Matt.